So next subtopic in polymerase chain reaction is its different components. PCR, we know, can generate 100 billion different copies of a specific DNA sequence in a matter of hours. And main components in PCR that include DNA template, sequence of DNA which is to be copied, primers, specific sequence of DNA which attaches in the DNA template at specific site, enzymes, tag polymerase, then new nucleotides which are D and TPs, which are required, different buffers, and different cations, for example, magnesium, manganese, and potassium ions. So the first component is a DNA template. We already know it is double-stranded and double helical molecule. There are two strands in it. One strand is in 5' to 3' direction, other is 3' to 5' direction. These rungs, they are formed of nitrogenous bases. Adenine base in one strand is attached with thymine on the other strand, and guanine base in one strand is attached with cytosine on the other strand. Then we have primers. What are primers? Primers are short pieces of DNA which serve as starting point for DNA synthesis at DNA template. The DNA synthesis starts at 3' end of the primer. Since the primers are custom built, they can have any sequence of the nucleotides which depend on the type of gene which we want to study. In a PCR experiment, two primers are designed. One is forward, other is reverse, which match to the segment of the DNA which is to be copied. Then primers and temperature requirement. Temperature chosen for a PCR experiment are determined by the primer sequence and the requirement of the DNA polymerase which is being used for the experiment. Annealing temperature which is used, that is normally between 50 to 56 degrees Celsius, but this selection has to be correct, otherwise it may affect the result of the experiment. If temperature is too high, then primer may bind to some other DNA region, and if the temperature is too low, primer may not even bind with the DNA at specific site. Hybridization temperature is determined largely by A plus T and G plus C composition of the primers. G plus C base pairs, if they are present in more in number, then temperature required is high one, and if A plus T base pairs, they are in less number, then the temperature, of course, is less as compared with if G plus C pairs, they are more. Ideal temperature for hybridization are calculated, which is based on G plus C and A plus T percentage in the primers, and accordingly, the hybridization temperature is calculated. Hybridization of primer with DNA template, that depends on the similarity of their sequence. What is DNA polymerase? DNA polymerase is an enzyme which binds with primer template and starts formation of new DNA molecule. DNA polymerase, which is generally used, is derived from a strain of bacteria which is called Thermus aquaticus, and that is why it is named as tag polymerase. These bacteria can survive in boiling conditions, in boiling temperatures, for example, even temperature of 72 degrees Celsius. And this results in the duplication of the original DNA molecule. Nucleotides, which are required for synthesis of new DNA molecules. What are these? Nucleotides are building blocks which make up new DNA molecules. There's a mixture of 4 types of nucleotides which are required, which are adenine nucleotide, cytosine, guanine, and thymine. DNA polymerase grabs the new nucleotides and attaches them to the end of the primer, and in this way new nucleotides are added, and in this way new molecules are synthesized. Extension time depends both on DNA polymerase which is used and the length of the DNA fragment which is to be amplified.